All right, we're diving into the final, absolutely critical chapter in the story of life's code. It's all about how DNA replication, this massive copying process, knows exactly when to stop. This is the grand finale, termination.
Just think about this for a second. Inside every single one of your cells, a story is being written, a story that's 3 billion letters long. And it's not just happening once, it's happening over and over, right now, inside of you, with this unbelievable speed and precision. It's the very essence of life, isn't it?
But here's the big question. How does that story know exactly when to end? I mean, if the copying machinery just kept on going, it would be total chaos, a genetic disaster. So how does the cell know it's reached the end? Well, that's what we're here to figure out. The answer lies in a process called termination.
Okay, Section 1, Replication's Grand Finale. Let's really set the stage here. You can think of DNA replication like a 3-act play, and termination? That's the show-stopping final act.
So what is termination exactly? At its core, it's the biological stop signal. It's this whole set of incredibly precise mechanisms designed to do one thing: slam the brakes on the replication machinery the very instant the entire DNA strand is copied. Not a second too soon, not a second too late. And this really helps put it all in perspective, right? You've got initiation, that's the launch. Then you have elongation, which is the massive construction phase. And then you have termination, which comes in and seals the deal. It's that simple. Start, build, and stop. Perfect. Section 2, why hitting stop matters. So you might be thinking, why is this so important? Well, because in the world of DNA, a sloppy ending is simply not an option. The stakes are unbelievably high. I mean, just look at what can go wrong. Without a clean, precise stop, you're looking at a recipe for disaster. You could end up with incomplete copies of your genome, which is catastrophic. Or the machinery could just keep going, causing harmful mutations. The ends of your DNA could just fray, get degraded, and you'd lose essential genetic info. So getting this right is everything. It's about maintaining stability and keeping the whole cellular operation running smoothly. Section 3, 2 blueprints for termination. Okay, so how does life actually do it? Well, it's pretty fascinating. There's not just one single way. Life has come up with 2 totally different master plans for this, depending on who you are. So you've got 2 main strategies here. On one side, you have the prokaryotes. Think bacteria. Their DNA is a circle, so they use a pretty straightforward system, basically a protein roadblock that says you shall not pass. But then you have us, eukaryotes. Our chromosomes are linear. They have ends. And that creates a completely different kind of problem, a puzzle that needs a much more complex solution, involving protective caps and a special enzyme to maintain them. All right, let's dive into that first blueprint, the prokaryotic stop sign. This is the beautifully simple and super efficient system that bacteria use. The best way to picture it? Imagine a circular racetrack with a perfectly placed traffic stop.
So here's how it plays out. The replication machinery is just zipping around that circular DNA. It keeps going until, bam, it hits a specific sequence called a pterocyte. That's the signal. Waiting there is a protein called Tus, which acts like a bouncer, physically blocking the path. The 2 replication forks eventually collide and everything grinds to a halt. Then a little cleanup crew comes in. Enzymes like Topoisomerase untangle the 2 new DNA circles, and DNA Ligase seals up the final gaps. It's so elegant, so clean.
Okay, now let's talk about our system. And it's designed to solve a much, much trickier problem. See, our chromosomes aren't neat little circles. They're long lines, and they have ends. The big problem with copying a line is that you always risk chopping off a tiny piece from the end each time. It's called the end replication problem. To solve it, eukaryotes came up with an ingenious solution, a protective shield. So, in our cells, replication starts in a bunch of places at once. The replication forks just copy DNA until they run into each other and stop. That part's simple. But the real magic, the key to the whole thing, happens at the very ends of the chromosomes. These ends have special protective caps called telomeres. They're just like the little plastic tips on your shoelaces that keep them from fraying. And there's this incredible enzyme called Telomerase, whose whole job is to maintain these caps, to add a little bit back on each time. Once that's done, good old DNA Ligase comes in and seals everything up. Section 4, the molecular toolkit. So who are the star players in all this? Let's take a closer look at the molecular tools that get the job done.
Okay, this toolkit is pretty amazing. You've got your specialists. First, there's the Tau protein. That's the dedicated roadblock, only found in prokaryotes. Then you have telomerase, the absolute hero for eukaryotes, the guardian of our chromosome ends. And then you have the all-rounders, the ones that work everywhere. Topoisomerase is the master detangler, a molecular pair of scissors. And DNA Ligase? That's the universal superglue, sealing up the final gaps in pretty much all forms of life.
Section 5. The perfect finish. Whew, okay, we have covered a lot. So let's wrap this up and make sure it all sticks with a couple of really simple memory tricks.
You gotta love this one. For prokaryotes, how do you remember it? Simple. Tus stops the bus at the Ter stop. The Tus protein literally stops the replication bus right at the Ter site. Perfect, right?
And what about us eukaryotes? Just think. Telomerase is the tail protector. It's the enzyme that protects the vulnerable tails of our linear chromosomes. It's our own personal shield, making sure nothing important gets lost.
So here's the bottom line. Termination is not some minor detail. It's an absolutely essential final step. Prokaryotes do it with Tus and terasites. Eukaryotes, like us, do it with telomerase and telomerase. But no matter how it's done, the goal is always the same. Accuracy, stability, and ultimately the survival of the cell. Because in the end, that's what this is all about. This entire beautiful, intricate dance has one purpose: to make a perfect copy of the instruction manual for life, every single time. No mistakes allowed.
And that leaves us with a pretty mind-blowing thought, doesn't it? We know that the shortening of our telomeres, those little protective caps, is directly linked to how our cells age. So, if telomerase is the very thing that protects them, could unlocking its secrets give us a key to slowing down aging itself? It's a huge